Okay guys, let's talk about enzymes. It's a little biological worker that makes the reaction happens faster, but they're also very picky about their working conditions. So in this video, I am going to talk about 3 things, so stay tuned. I'm Top GCSE, and if this helps, hit subscribe so revision stops feeling like a punishment. Thank you. Right. So first, what actually is an enzyme? It's a biological catalyst, meaning that it speeds up the reactions but doesn't get used up. Like that one friend who's always organizing the group chat but never leaves it, technically. Enzymes have an active site, a specific 3D shape where substrate fits. You might hear lock and key, where the shape matches perfectly, but a nicer explanation is induced fit. Okay, the active site can slightly mold around the substrate to get the perfect fit. So the key idea here is shape matters, and if the shape changes, the enzyme basically can't do its job technically. Do you know what I mean? Now let's have a look at temperature. Temperature is classic exam favorite. So this is so listen to this like it's paying rent. As the temperature increases, enzyme and substrate particles have more kinetic energy, so they move faster and collide more often. More collisions means more enzymes and substrate complexes, so the rate goes up. When you hit an optimum temperature, for human enzymes, it's often around 37 degrees Celsius, where the enzymes are working with maximum efficiency. Anything beyond, they just become denatured, it's just irreversible, so the reaction rate drops dramatically. So the graph story is up, peak, and then drop dramatically. Now the pH. Now basically, how acidic and alkaline something is, it affects their enzyme shape. Different enzymes have different optimum pH. Say for example, pepsin has pH 2, super acidic, it's in the stomach, and trypsin is small intestine, is in the small intestine, pH 8, more alkaline. So if the pH goes too high, too low, that also kind of disrupts some kind of same kinds of bonds holding the enzyme structures together. And at extreme pH, you can get permanent denaturation. So the enzymes become basically useless. So pH is like vibes, too acidic, too alkaline. Enzymes like, I'm not working in these conditions, kind of thing. Another one is the substrate concentration. At low substrate concentration, if you add more substrate, you get more frequent collisions, so the reaction increases. It's pretty like linear at first. But eventually what's going to happen is you reach a point where all active sites are occupied, meaning every single enzyme is already very busy. So the enzymes now become the limiting factor. Adding more substrate is not going to help. So the concentration of substrate helps to some point. It's like a checkout queue. Adding more customers doesn't make it faster if you still have the same number of cashiers, guys. Are you with me? I hope you are. Right, going on to structuring a 6-marker response on this. The key, this slide is gold, so take a screenshot of it, take a picture of it, pause it, make notes on it, whatever you want to do. Now, define the key term. Enzyme is a biological catalyst, it lowers activation energy. Explain collision theory. Discuss denaturation. Link to active site. The active site shape changes, so substrate no longer fits, fewer complexes. Apply to data. Mention things like trends, steep, drop, any given value that is there, and conclude your answer scientifically. Limiting factors. State optimum levels. Rate decreases due to it being denatured. Now, if you can do this in a calm, logical chain, you're basically printing marks, guys. If you want more of these how to answer 6-marker videos, Do not forget to subscribe. I am Talk GCSE and I have got you guys.